similarities, right? Your parents and you, between your parents and you, there are some similarities. And why that similarity is there? There is some reason now. And from your family and someone from the other family, there are difference. No? The difference is there. So why that difference is there? Okay. So the genetics will deal about the inheritance. The similarities is due to the inheritance of the genes, right? The genes are transmitting from the parents to the offspring. That's why there is similarities between you and your parents, or between your within the family. Okay, some common genes are transmitted, and you know that whatever you are, it's you look fair, this much tall, this and that. It's mostly due to the gene. Okay, the genes are responsible for whatever you looks like or whatever you have. So that is. But later on, you will come to know that there are some other components also for whatever you are. Okay, not only genes, there are some other factors also. Say, for example, say you in a farm, in a cattle farm, suppose a cow is producing, say, 10 liters of milk per day. Suppose a cow is producing 10 liters of milk per day. And some other day, it may produce 15 liters of milk per day. So what may be the reason behind it? Is it only the genetic component which is responsible for the 10 liters of milk or 15 liters of milk? That is the question. Okay. So am I audible to you?
Yes, sir.
Okay, good. So that is like, not suppose the animal is there. It's a very good animal, genetically very strong animal. Okay, it has the capacity to produce, say, 20 liters of milk. It has the potential. The genetic makeup is there. Remember, and the genes are responsible for the production of milk. Okay, so it has the potential to produce, say, 20 liters of milk. If you are not feeding well, then the animal will produce that 20 liters of milk or not. That is the question, right? If the management is very poor, and if you are not feeding the animal properly, then the animal may not produce up to the optimum, say 20 liters of milk. Okay, so, and moreover, say for example, what happens is, you know the Jersey, have you seen Jersey, yeah, Holstein Friesian cattle? Jersey, yeah, Holstein Friesian cattle, they are the exotic cattle. No, you have seen, no, that Holstein Friesian cattle, that
Yes.
white patches, the black,
Yes, sir.
yeah, those are the animals from the temperate region, no, from the temperate region. So when they are imported here, so you might have seen in your locality, no? So when they are imported here, they are not producing up to the optimum. Okay, so in that temperate region, the animal may be producing 20 liters of milk, but the same 20 liters of milk, if you import the animal here, it may not give you 20 liters of milk, because your environmental temperatures and all those conditions are quite different there. Our is tropical region, quite hot, humid, and it will be very stressful to those temperate region animals, right? So that's why for any phenotype, you know what is phenotype? There are two things. One is genotype and the phenotype. Okay, the genotype is about the genes, and the phenotype is whatever you are seeing. Okay, whatever you can measure. So animal is producing 20 liters of milk, means that 20 liters of milk, that is the phenotypic expression of the genes, right? So To produce 20 liters of milk, one thing is there should be the genes responsible to produce that milk. Okay, not only genes, there is other factors like that is your uh, the environmental factors. Environmental factors. Okay, so to produce your 20 liters of milk, you need the genotypes, the genes responsible for production of that milk, as well as the environmental factors. Environmental factors, it may include all the non-genetic factors. Non-genetic factors means the environment, the temperature should be this much, this much. Okay, there should be some optimum temperature. You know? If the temperature is very hot, then it will be stressful to animal, and it may not give you, the animal may not give you that optimum number of the milk. Okay, then the feeding also. The feeding has to be optimum. Okay, so like that, there are two components. One is the genotype and the environment. Okay, so like that. Sorry. So genetics, the genetics deals with two things. One is the inheritance and another is variation. Okay, why there is variation between the individuals? You and your neighbors or your friends from the different families, they are different. There's variation. Okay, there's variation between in the individuals to individual. Okay, so these two components will be studied under the genetics. So if somebody asks you, don't forget to mention these two things. One is the genetics will deal with the inheritance of the characters or the genes and the variation among the individuals in the population. Okay, the inheritance and the variation. So why there is similarities and dissimilarities? Simply that is similarities and dissimilarities. Similarities is the inheritance and dissimilarities is the variations. Okay, so don't forget. Okay, then let us see about the history of genetics. You know what is genetics now. The genetics will study about the inheritance and the variation, why there is variation and why there is similarities. So in the earlier time, wait. Yes. Wait. Wait, the skill, the slide is not changing. I'm trying to change. Still, you can see the slide, right?
No, sir.
There is some problem going on.
No, sir.
Okay, some problem is going on. The slide is changing. It's not changing.
Sir, I think you need to go to the presentation settings.
No, the presentation is not taking here. It's not coming. No, that's why.
So
Actually,
there
where?
are two different pages showing it. one is your face and another is the presentation. I think you need to go to the presentation slide and go to present now option down below.
Present now, resume presentation. Resume presenting.
Yes, sir, right there.
It's coming up.
Yes, sir, yes, sir.
But the thing is, it's not taking next slide. Actually, I'm using the Mac, no? That's why actually this PowerPoint is Mac and this one Microsoft.
Oh.
That's why in the Mac, no, it's.
So I think for now you need to go to the PowerPoint app itself and then do the sliding thing.
Okay, let me see. So can you see the screen now? Can you?
Yes, sir.
Yes, sir.
Yes, sir. The history of genetics is coming, is it?
Yes, sir.
Yes, sir. Yes, sir.
No, it's okay then. Okay, anyway. So as I told you, there are two things in the genetics. One is the inheritance and another is the dissimilarities, that is the variation. Okay. So the people have known about inheritance for a long time. Say, you know that children resemble their parents, and the domestication of the animals and plants, the selective breeding of the good characteristics has been going on since long. Okay, you can see that this slide. Can you?
Yes, sir. Yes, sir.
Okay then. Like from the earlier time, so people used to select the animal on the basis of certain good characteristics, right? So and the breeding is going on. So they select the animals, the best animals, the male and female, and the breeding used to be. So since time immemorial, these things are going on. Okay, then there is the earlier this one, the Sumerians. They have the horse breeding records. Okay, they kept the records of the horse breeding. Then they have the ability to identify a person as a member of a particular family by these certain physical traits. Okay, so even like earlier, so people can identify a person as a member of a particular family by certain physical traits. Okay, these things are all going on. So you can say that the people are knowing about the inheritance for a long time. Then there's a lot of ideas, all ideas, the conflicting or maybe some good ideas also. Despite knowing about inheritance in general, a number of incorrect ideas had to be generated and overcome before modern genetics could arise. Okay, the modern genetics. So you know about the Mendel. So Mendel we used to call as the father of the modern genetics. No? Most of the theories of the genetics, it comes from the Mendel, okay, from the Mendel's era. So before that also there are a lot of theories here and there, okay. And you know that all life comes from other lives. living organisms are not spontaneously generated from non-living materials? See, spontaneous generation. What happened is, in the case of the spontaneous generation, there was like when you keep a piece of meat outside, okay, just keep a piece of the meat outside. So after a few days, you'll find some maggots are there, right? You'll find some maggots on the meat. So from that, the people earlier, they believed that the life originated from the organic decaying materials spontaneously. They don't believe on life comes from other life. Okay, they don't believe on life comes from other life. So by seeing the maggot on a piece of the meat, they believe that life originated spontaneously. Okay, so that was the belief. Then there are certain other concepts like species concept. Offspring arise only when two members of the same species met. Then the monstrous hybrids don't exist. Okay, there are a lot of concepts that were there. Then organism, some concepts are there, like organism developed by expressing information carried in their hereditary material. Organisms developed by expressing information carried in their hereditary material, as opposed to pre-formation. So pre-formation, what the pre-formation says is like the Leeuwenhoek. Have you heard about Leeuwenhoek? Have you heard about the Leeuwenhoek?
Yes, sir.
Yes, sir.
Yeah, you discover what? Microscope, right? So he and some of his followers believe that the living things, already a miniature figure, okay, of that individual is already there in the sperm or egg. Okay, already is there. The miniature. When looking for the sperms in the under the microscope, now he saw some spots, some figures like human being, this and that. No? So they believe that already miniature structure is there in that the sperm or the egg. Just 1 minute, okay. So that is the theory of pre-formation. So the pre-formation says that already pre-formed structure of the individual is there in the sperm or egg. Okay, that is the theory of pre-formation. Then the environment can alter the hereditary materials in a directed fashion. There is no inheritance of the acquired characteristics. So the inheritance of the acquired characteristics, this theory was given by Lamarck, no? Remember? So it says that you acquire something. Suppose you are doing the gym every day, so then your muscles will develop. No? Your muscles will develop. So those things will be inherited in your progeny or not. Okay. So those theories are there. Then there is like the male and the female parents contribute equally to the offspring. That is true, right? So 50% from the male and 50% from the females. So in the ancient Greek, they believe that the male plants a seed in the female garden. Okay, they believe that the female is not contributing; they are just a garden. The male will plant a seed in the female. The female is taken as the garden. They will nourish the seed. Okay, that was the belief. Then in the New Guinea, they believe that sex is not related to the reproduction. Okay, these were the old thinkings. Then comes the Greek philosophers. Theophrastus proposed that the male flowers cause female flowers to ripen. Okay, he believed that these male flowers are responsible for the ripening of the female flowers. Then the Hippocrates, the Greek scientist, the Hippocrates, speculated that seeds were produced by various body parts and transmitted to offspring at the time of conception. Okay. He speculated that the seeds were produced by various body parts and transmitted to offspring at the time of conception. Yeah, this was the theory of Hippocrates. Then Aristotle, he thought that male and female semen mix at conception. Okay, male and female semen mix at conception. This was the thinking of the Aristotle. Then Achilles proposed that the male as the parent with the female as a nurse for the young life shown within her. Okay, Achilles believed that male is the actual parent, but the female is the only, is the nurse for the young life shown within her. Okay, that was the belief. Then there are the theories like the blending theory. So the blending theory comes like the mixture of the sperm and the egg resulted in progeny that were a blend of two parents' characteristics. Okay, the sperm and egg they blend, and the progenies are the mixture of those the two parents. Okay, the male and female parents. Then you have the acquired characters inheritance, that is Lamarck's theory. The individuals inherit traits that are strengthened by their parents. So there is, suppose some individual's parents is doing some say gym, is going to the gym regularly and has developed a big muscles. Okay, biceps, triceps, all those things. So those things will be inherited to the progeny. That was the thinking of the Lamarck. Then Charles Darwin, he have given one theory that is pangenesis. Okay, what the pangenesis says is each part of the body continually imitate its own type of small organic particles. See, each part of the body continually imitate its own type of small organic particles called as gamules. Okay, gamules that aggregated in the gonads, that is in the reproductive parts, contributing heritable information to the gametes. Okay, so it's according to the pangenesis given by the Charles Darwin, it said that each part of the body or each tissue of the body, it produces small invisible organic particles, that is gamules. Okay, that is we call as the gamules, and these gamules or pangenes, sometimes also called as the pangenes. These gamules are transferred by the circulatory system to the reproductive system, that is in the gonads, and this will be transmitted these gonads. Okay, and from these parts, the small gamules, the small gamules are believed to be the representative of each of the body parts. So the individual, the progeny, the individual will be developed from those small gametes. Okay, the different parts of the body will be developed from that small gametes. Okay, that was the belief of the Darwin. Okay. Then there was pre-formation and epigenesis. As pre-formation, I told you, there is only the pre-formed structures are there in the sperm or in the egg. So there are two groups of the people. One is we call as the animalculists. So animalculists, they believe that these pre-formed structures, miniature structures, are present in the sperm. Okay, the sperms is responsible for the development of the individuals. They believe it. And some people, they believe that they are called as ovis, okay, ovis, O-V-I-S-T, ovis. They believe that egg is the responsible for the development of individuals. So while looking for the eggs, the ova, they found some structure in the ova. So they believe that only the miniature or the pre-formed structures are present in the egg or the ova. Okay, so there are two contrasting thinking. So one belief that the sperm is responsible for the development of the individual, and another belief that egg is responsible. So those two thinkings are there. Pre-formation and epigenesis. So structures arise during development that are not only the pre-form. So that contrasting thinking is like in the free-form, it says that only the structure is there. But the epigenesis, the theory of epigenesis says that the different parts of the organisms, different parts of the individuals, they are developed from undifferentiated structure, okay, at different stages of development. See, pre-formation theory says that already the full structure of the individuals are there. And the epigenetic theory says that it is not like that. The structures of the different parts of the individuals are different from some undifferentiated structures at the different stages of the development. Okay, so that is the epigenesis. Then the cell theory is there. The cell theory, it says that all living things are composed of one or more cells. All living things are composed of one or more cells. It's true, right? Then cells are the basic unit of the structure and function in living things. You know that cells are, some are trying to join. Let me try to see. Where is it? Admit. Then these cells are the basic units of structure and function is given by the skeleton and schwann. Okay. Then new cells are produced from the existing cells. So it's given by the Virchow. Virchow says that the new cells are produced from existing cells. Then in mid 1800s, there are so many discoveries are there. So, like in 1859, Charles Darwin published The Origin of Species. Origin of Species was published by Charles Darwin in 1851. Okay, it described the theory of evolution by natural selection. It's a very important book in the evolution. Okay, the theory of evolution by the natural selection. Then the Gregor Mendel, as I told you, he is the founder of the most of the theories of the genetics. So he published his experiment. He published experiments in plant hybridization in 1866. Okay, he have done some meticulous work. He have done the meticulous work on the pea plant, and meticulously his good in the mathematics also. Okay, so that's why the data are collected nicely, systematically, and he did the analysis, so he could understand the things there. So then he published his work in the that is experiments in plant hybridization in 1866. And interestingly, that time no one understood his theories, okay, his writings. So almost up to 1900, nobody understood his findings. Okay, only in 1900, three scientists that we see later on, they rediscover independently. They rediscover his findings, and it found to be a very rigid finding. Okay, so all the theories of the genetics is going to start from there. Then in 1871, the Frederick Richter, he isolated the nucleic acid from the pus cells. So, okay, remember. So DNA, the double helical structure of DNA was discovered in 1953, right, by the Watson and Crick. But this nucleic acid they have understood long time back in 1871. 1871 to 1953, see, the structure developed in 1953, but in 1871, the already nucleic acid has been, someone is trying to join. 16, no. Okay, 63. So that is, okay, Gregor Mendel, regarding the Mendel, we will discuss separately. I will take it aside for time being. Instead of that, we will see these things we will discuss later on. Okay, this is in the nutshell. We will see these things, scientists and their work. In 500 BC, okay, long time back, Pythagoras has given the vapor fluid theory. Vapor fluid theory. Okay, actually, this slide now all looks very consistent. I make these things in the Microsoft, and when I transfer here in the Mac Apple, so it's all this one setting has gone. Okay, so next time I will take in the window only. So all the setting has changed. Someone is trying to join, I think. All the setting is right. No. So, am I audible? Are you hearing me?
Yes, sir.
Yes,
sir.
Yes, sir.
Okay.
Yes, sir. Crystal clear.
Okay, good then. So, Pythagoras has given the vapor fluid theory. So, what the theory says is a certain moist vapor is transmitted from the male. Okay, a certain moist vapor is transmitted from the male towards the female during the fertilization, and it developed into an embryo. That was the belief of the Pythagoras. Okay. Vapor fluid theory means a certain moist vapor is transmitted from the male towards the female during the fertilization, and it developed into an embryo. Okay, that is. Then in 250 BC, Aristotle suggested that semen is highly purified blood. Semen is highly purified blood. So Aristotle believed that. Then William Harvey mentioned that eggs give rise to living things. So Harvey is the. So I told you at that time there are two thinkings. One is anemic police. Those believe that egg gives the life. And another group is Ovis, O-V-I-S-T, Ovis. They believe that egg give rise to the living things. Okay, that's those two thinking. So this William Harvey is one of those obvious. He is mentioned that egg give rise to living things. Okay. Then in 1977, Liu and Hawk, it shows the living sperm in the semen of many animals. He discovered a microscope. Then he started looking for the sperms in the semen of many animals. Then this Swammerdam it suggested that the sperm alone furnish the. Okay, this is animepolis. So he believed that sperm alone furnish the embryo, while the uterus of the female is necessary for the enlargement of the embryo. So thus, only the uterus of the female gives the nourishment to the sperm. Okay, that was the the thinking of the Swammerdam. And he's the first person to do AI in the dog. Okay, Swammerdam is the first person to do AI, artificial insemination, in dog. And then graph is also obvious, suggests that it is the egg, not the sperm, which contain the preformed embryo. Swammerdam is animepolis. We believe that in the sperm, the preformed structures are there. And graph, admit Sarah. Then the epigenetic theory, as I told you, what is epigenetic theory? The epigenetic theory, this Swammerdam, graph, and these things, no, they believe on the preform theory. Okay, the preformation theory. Already the miniature structure of the individuals is there in the sperm or egg. Okay, they believe on that. That is we call as the preformation theory. Then this Wolf proposed this. Yes. And Wolf proposed the epigenetic theory. The epigenetic opposed the preformation theory. As I told you, in the epigenetic theory, the parts of the body are different, are developed, evolve, or develop from the undifferentiated structures at the different stages of development. No, that is epigenetic theory. It's given by the Wolf. Then Lamarck put the theory of acquired characters in the theory of acquired characters. Acquired characters says that if something occur in your life, no, occur physically, then those things will be inherited to your progeny. That was the belief of Lamarck. But many scientists have disproved it, have countered the theory. Then Charles Darwin proposed the theory of pangenesis. And as I told you, what is pangenesis? Every part of the body, the tissue of the body, they produce some invisible small particle that is we call as the gametes or pangenes as the representative of that part, body part. Okay, so those things, the gametes are transported to the gonad, the reproductive systems, to the blood circulations, and from that the living things develop. Okay. Then Hardwick in 1875 discovered the fertilization of male gamete and the female gamete. Then Fleming is very important, no? Fleming discovered the process of splitting of chromosomes. Okay, the process of splitting of chromosomes and their movement to the poles. Okay, this was discovered by the Fleming. That is, in the cell divisions, the homologous chromosomes. You know that the the chromosomes are present in pairs, no? In pairs. So these homologous chromosomes, they split, okay, they separated and they move to the opposite poles at the time of the cell division. Okay, that was discovered by Fleming. And also discovered the Lambrus chromosome. Lambrus chromosome is we call as the giant chromosome. It's a very big chromosome. Okay. So what is Lambrus chromosomes? Those things we'll see later on. Then Beneden suggests that gametes are haploid, while somatic cells are diploid. Haploid and diploid, do you have any idea? Haploid and diploid. Remember, twice N and N. Single N.
Yes, sir.
Yeah, that is we call as the haploids. Not the gametes, your eggs or sperms are the haploid, not only N numbers. Suppose the human being has how many chromosomes? How many numbers of chromosomes?
46.
46. That means that is the twice N, no? That is the twice N. That is the diploid numbers of the chromosomes. Okay. So then the N is 23 numbers of chromosomes. Okay, these 23 numbers, they are present in pairs. Okay, say these 23 numbers of chromosomes are all different. All different. Someone's trying to join. See, if somebody asks how many types of the chromosomes are there in human being, generally there are two types of the chromosomes. One is autosomal chromosomes and another is sex chromosomes, right? Sex chromosome, your X and Y, and the others are we call as the autosomes. Okay, but the types of the chromosome, if we say, that is in general. Okay, types of the chromosomes, if you say the chromosomes, we call as the karyotype. Okay, karyotype, Karyotype, karyotype. So what is karyotyping is we arrange the chromosome linearly according to the shape and size. Okay, according to the shape and size, we can arrange the chromosomes. So when we arrange the chromosomes, so what we found is there are 24 different types of the chromosomes in the human being. 24 different types. So chromosome 1 is biggest. Chromosome 1 is biggest. So chromosome 1 has pairs. Okay, there will be two chromosome 1s. Then chromosome number 2, it will also be present in pairs. Okay, there are two two similar chromosomes. Okay, of chromosome number 2. Like that up to 23 pairs. 22 pairs. 22 pairs, okay. So 22 autosomal pairs and 1 sex chromosome pairs. So these sex chromosome pairs may be of the homologous type. Homologous type means the same type or maybe of different type. So same type is, in the case of the female, you have XX. So they have same pair. And in the case of the male, you will have X and Y. X and Y, their shape and size are totally different. Okay. Someone is trying to join. No, it's not. So, like that. What was that I was saying? The gametes are haploid and the somatic cells are diploid, that is. Then the term chromosome was coined by W. Waldeyer. Okay, the term chromosome was coined by W. Waldeyer. So chromo, when we dye the chromosome with some basic dyes, okay, it takes the color. Okay, that it takes the color. So that's why chromo means color. Soma means the body. So it is called as the color body. Chromosome means the color body. So chromo means color, soma means body. So when we dye the chromosome, it takes the color, okay, with the basic dye. That is, so the term chromosome was given by Waldeyer. Then the Weismann gave his own theory of continuity of germplasm, the theory of germplasm. So the theory of germplasm, it says that the body has two types of these cells. Okay, two types of these systems or the cells. One is we call as the somatoplasm. Another is germplasm. Somatoplasm, soma is body, no? The somatoplasm and the germplasm. So the germplasms are like the reproductive cells. Okay, germplasms are your gametes, maybe eggs or sperm cells. They are we call as the germplasm, and other cells are we call as the somatoplasm. Okay, body cells. So these are two things, and and according to the theory of germplasm, only this germplasm, not the somatoplasm, is responsible for the inheritance. Okay. Say, suppose if you cut your hand, okay, you just cut in your hand. So what will happen? Your son or daughter will still have the hands. No? Your son or daughter will still have the hand. Okay, if you cut your hand. Okay, that's what. This somatoplasm is not involved in inheritance. Okay. Whereas in the case of this, you're seeing the screen, right?
Yes,
Hello?
sir.
Yes, sir.
Yes, sir.
Sorry. This is not the one. So, but if there is something wrong in the germplasm, egg cell or sperm cells, okay, that defect will be carried on to your offspring. Okay, if there is something wrong, some defect is there in your egg cell or the sperm, so that will carry on to the offspring. Okay, that is we call as the theory of germplasm. It's nothing but the somatoplasm, the body cells, they are not involved in the inheritance. Only the germplasm, germ cells are responsible for the inheritance. Okay, that is. Then Landsteiner, it gives the concept of the blood groups in the human being. Okay, remember this is important. The blood groups in the human being was given by the Landsteiner. Then again, in 1900, I told you in 1866, Mendel has published his finding, no? The experiments in plant hybridization. So his findings, no? Nobody could understand for so long, almost for 40 years. But only in 1900, these three scientists, Correns, Hugo de Vries, and Semark, the three independently rediscover his finding and someone is trying to join. And they prove that the Mendel's finding principles are valid. Okay, only in 1900. Then in 1901, the term mutation was coined by Hugo de Vries. Mutation was given by Hugo de Vries. Okay. Then in 1902, described the presence of sex chromosomes. Then Sutton has given the chromosomal theory of inheritance. Then the term genetics was given by Pattison in 1902. Okay, the term genetics was given by Pattison. He also gave the terms like homozygous, heterozygous, F1 and F2 generations. These terms were given by the Pattison. Remember, okay, the term genetics was given by Pattison, not only the genetics, homozygous, heterozygous, the F1, F2. What is the full form of F? Anyone? What is F?
Filial.
Yeah, first filial, second filial, like that. Okay. Then the term gene, genotype, and phenotypes. Gene, genotype, phenotype were given by the Johansen, okay, in 1909. Then Morgan, Thomas Hunt Morgan, he did extensive work on Drosophila. Drosophila is your fruit fly. No? In Manipuri is called tumit, the fruit fly, and gave the concept of sex determination and sex linkage. Okay, the sex determination and sex linkage, this term was given by Morgan. Then Breest has given the genetic balance theory of sex determination. Genetic balance theory of sex determination. Then the chromosomal abnormalities and aberrations. Okay, this was given by the Breest. So the genetic balance theory of sex determination, what is that? Is the female has 2 eggs and male has only 1 egg, right? So what happened is in the female, one of the 2 eggs is totally inactive. Okay, only 1 egg is active. That is we call as genetic balance theory. Why only 1 egg is active and another is inactive is to balance with the male. The male has only 1 egg. So like in some abnormal condition, there may be 3 eggs, triple X in the females. In some abnormal condition, those things we'll see in the chromosomal aberration. Okay, so the active X chromosomes are always one less than the total number of the chromosomes. Okay, that is we call as genetic balance theory. Then the multiple alleles in the blood group concept was given by Bernstein in 1925. Multiple alleles, do you know what is that? If a gene has more than 2 forms or more than 2 alternative forms, okay, then it is called as the multiple alleles. The example is the blood group. Blood group has your A, B, O, like that, no? They have different many alleles, no? So that is we call as multiple allele. It's what and it was given by the Bernstein. Then Muller artificially induced the mutation by the treatment with X-rays. Okay, so this is very important. So mutation, the artificially induced mutation by the X-rays was done for the first time by the Muller. Then let's see. Then RS factor in the blood was given by Landsteiner and Wiener. Then the Biddle and Tatum has given 1-in-1 enzyme theory, right? Something is trying to tell you. Then so before Watson and Crick in 1953, they discovered the double helical model of DNA. Some of the prerequisite things about the DNA was given by many. This one, the Sargaf has demonstrated that the DNA that was in 1950, the Sargaf theory model. He says that adenine, the amount of adenine is equal to thymine. Then cytosine, the cytosine is equal to the guanine. So these things has been given by the Chargaff already. Okay. Then Then in 1870s, all it and Crick. This Crick is the British scientist out there at the time. So Watson is American. No? Watson is quite young at the time. He was around 23, 24 only. He has finished the PhD also. And Crick was quite old, 36 or something like that. And actually, Crick is from the physics. Okay, he did his study on physics, degree and everything on physics. And Watson he did on biology. So she then tell me they met on a one conference, and so looking to their interests, so they come together in the King's College London. Okay, they started discovering the double helical structure of the DNA. So that time, what happened in the King College London, there were two other fellows. That is Wilkins and Wilkins is there, and another is Rosalind. The question is they have to pattern it. No? We don't say that Crick and Watson model. No? We say Watson and Crick model, right? We always said that Watson and Crick double helical model of DNA. We never said the Crick and Watson model of. So they decided how to give the name of this model. So what they did is they toss a coil. Okay, they toss a coin, and finally they have given it as Watson and Crick model, not Crick and Watson model. Okay, it was like that. And they got the Nobel Prize. The three fellows, Watson, Crick, and Wilkins. These three fellows got the Nobel Prize in 1962. But your Rosalind Franklin, she did not get it. So there are a lot of controversies there. One belief was, one thinking was like, so they have to share the money. So if there are four persons, it may be difficult to share the prize monies. Okay, so up to three persons only were given in there. Okay, that's why only two of them were given. But other theory is like this one Nobel Prize is not given posthumously. She died very early, in 32 years of age. Okay, before that announcement of Nobel Prize, Rosalind already expired. Okay, so till 1974, posthumously the Nobel Prize was not given. Okay, that's why it may be she did not get the Nobel Prize. Then Kronberg They discovered DNA polymerase of E. coli, then Corona and Nuremberg awarded the Nobel Prize for completing the genetic code for the simple enzyme. Genetic code for the simple enzyme. Oh, I have to rush off. Your class is there from 11:15, right? Nutrition class.
Yes, sir.
And finally,
Yes, sir.
McClintock got Nobel Prize for his discovery of the jumping genes. Have you heard about the jumping genes? Or sometimes we call it the transposable elements. So what happened is,
Yes, sir.
in the genome, there are some genes, some part of the DNA, which position is shift or be changed very frequently. Okay, some part of the DNA. So it may be one day it may be here and one day it may be in another chromosome. So they may shift their position within the same chromosome or to the other chromosomes. Okay, so those parts of the your genomes are we call as the jumping genes. Okay, jumping genes. They just shift, change their position within the same, maybe within the same chromosome or to the other chromosomes. That is we call as the jumping genes or the transposable elements. So it was discovered by the McClintock. Okay, that's all for today. We'll see in the next class. Okay, if there's any problem, tell me. Is anyone? You want to ask anything? So silent. Okay, bye for today then.